Assalamualaikum. Hi everyone. Today we'll be discussing polymerase chain reaction, or in short PCR. PCR is a widely used technique in molecular biology that allows scientists to produce many copies of a specific double-stranded DNA segment. It works by mimicking the natural process of DNA replication, but it takes place entirely in vitro. The PCR process relies on precise temperature manipulation to control each step of DNA amplification. PCR uses a series of temperature changes to drive three main steps: denaturation, annealing, and extension. These steps are repeated over multiple cycles to produce large quantities of DNA, known as amplicons. So how does PCR work? In a PCR reaction tube, there are four essential components: the template DNA, a pair of primers, one forward and one reverse, DNA polymerase, and the four building blocks of DNA, which are deoxyadenosine triphosphate, deoxyguanosine triphosphate, deoxycytidine triphosphate, and deoxythymidine triphosphate. These components are mixed together in PCR buffer solution to create the right environment for the reaction. In the first step of PCR reaction, the denaturation process begins when the temperature is increased to 94 to 98 degrees Celsius, causing the double-stranded DNA, which serves as the template in the reaction, to separate into single strands. Next, primers anneal to the complementary sequences on the template DNA strands. This process, called annealing, occurs at a temperature of 50 to 65 degrees Celsius. The two different primers are short sequences consisting of 18 to 25 nucleotides, and they serve as sequence-specific starting points for DNA synthesis. In the third step, the temperature is raised to 72 degrees Celsius, where DNA polymerase binds to the primers and begins adding nucleotides to synthesize a new DNA strand. As a result, one double-stranded DNA molecule gives rise to two daughter strands, represented by the equation 2 to the power of n, where n is the number of cycles. By repeating these steps continuously, a specific segment of DNA is amplified exponentially. Typically, PCR runs for about 25 to 40 cycles, depending on the intended application. Even at the lower end, with just 25 cycles, a single double-stranded DNA molecule can produce more than 33 million copies within 1.5 to 2 hours reaction time. And that's how PCR turns heat and cycles into billions of copies, one strand at a time. Until next time, and thank you for watching. Assalamualaikum.